In 1921, a failed surgeon with no research experience, working in a borrowed laboratory with second-hand equipment, made the most important medical discovery of the 20th century. He sold the patent for $1 so that no one could ever profit from it. Insulin now costs $300 a vial in the United States. This is the complete story of how that happened.
You are standing in a laboratory on the second floor of the medical building at the University of Toronto. It is May 21. The laboratory does not belong to you. It has been lent to you, reluctantly, conditionally, for the summer only, by a professor named John MacLeod, who has gone to Scotland on holiday and left behind a junior assistant to keep an eye on what you are doing. You are Frederick Banting. You are 29 years old. You are a general practitioner from London, Ontario, whose practice has been failing. You are not a researcher. You have never conducted a laboratory experiment in your life. You have one idea. One single idea that you wrote down in a notebook at 2 in the morning on October 31st, 1920, after reading an article about the pancreas in a medical journal. The idea is this: the pancreas produces something, some substance, some secretion, that controls the way the body processes sugar. Diabetics do not have it, or cannot use it. And if you could isolate that substance, extract it, purify it, inject it, you could give diabetics what their own bodies cannot make. Every researcher who has had this idea before you has failed. The best biochemists in the world have been trying to extract this substance for 30 years. They have all failed. The substance degrades before it can be isolated. The digestive enzymes of the pancreas destroy it before it can be extracted. Every laboratory that has tried has hit the same wall. You have a different approach. You have written it in your notebook. Ligate the pancreatic ducts of dogs. Wait 6 to 8 weeks. The digestive cells will die. The substance, whatever it is, may survive. You do not know if this will work. You have never done it before. You have been given 10 dogs, a second-hand centrifuge, a borrowed laboratory, and until September, when MacLeod comes back from Scotland and reclaims the space. You have 4 months. You have 14 dogs by the time you are done. And you need to talk about what happens to them. Because the story of insulin is not just the story of discovery. It's the story of what discovery costs. Not in dollars, in something harder to quantify than dollars. But before we go into that laboratory, before we follow what happens in those 4 months, you need to understand what diabetes meant in 1921. Not what it means today, what it meant then. Because today, diabetes is a manageable condition. A serious one, but manageable. In 1921, diabetes was a death sentence. Not a slow one, not a comfortable one. A death sentence that was delivered to children, to teenagers, to people who had decades of life ahead of them, and whose bodies were consuming themselves from the inside out. Type 1 diabetes, the form that Banting was trying to treat, is an autoimmune condition in which the body destroys the cells in the pancreas that produce insulin. Without insulin, the body cannot process glucose. Without glucose processing, the cells starve. the body begins consuming its own fat and muscle for energy. The process produces toxic byproducts called ketones. The ketones accumulate in the blood. The blood becomes acidic, and the patient slowly, over weeks or months, wasted away. In 1921, there was one treatment for type 1 diabetes: starvation. Not metaphorical starvation, literal starvation. Dr. Frederick Allen at the Rockefeller Institute had developed what he called the starvation diet, reducing caloric intake to the absolute minimum that could sustain life while slowing the progression of the disease. It extended survival, sometimes by months, occasionally by years, but it did not treat the disease. It simply slowed the pace at which the body destroyed itself. The wards where Allen's patients were kept were described by every physician who visited them in terms that do not vary much between accounts. The patients were skeletal, children of 10 or 12 who looked like photographs of famine victims, adults reduced to 80 or 90 pounds, patients so weakened that they could not stand without assistance, patients who had been told by their doctors, honestly, because honesty was all that was left to offer, that they were going to die, that the only question was how long the starvation diet could slow the process. This was the context in which Frederick Banting arrived in Toronto with his notebook and his idea and his borrowed laboratory. This was what was at stake in the building on the second floor of the medical building at the University of Toronto. Not a research paper, not a career advancement, not a grant renewal. The lives of every person in every ward, like the ones Allen was running in New York. Banting knew this. He also knew that he had 14 dogs and until September, and that the most important thing he needed to do before any injection, before any experiment, before any extraction attempt, was solve the problem that had defeated everyone who had tried before him. How do you isolate a substance from a gland that destroys it the moment the gland is disturbed? His notebook answer was the pancreatic duct ligation. Kill the digestive cells, leave the insulin-producing cells, the islets of Langerhans, intact, then extract what remains. He began operating on the dogs in May 1921. By July, he had results. By August, he had something that looked like it might work. By September, when MacLeod returned from Scotland and saw what Banting had done in his borrowed laboratory with his borrowed equipment and his assistant, Charles Best, a medical student who had won a coin toss to spend the summer helping rather than going on holiday, MacLeod understood immediately what he was looking at. And what he did next is the part of the story that Banting never forgave him for. MacLeod was a skilled scientist and an experienced academic administrator. He understood how to recognize important research. He also understood something that Banting, who had never operated in an academic institution before, did not. He understood how credit worked, how it was assigned, how it was distributed, and how, in the politics of an academic laboratory, the person who controlled the resources had a legitimate claim to a share of whatever those resources produced. Banting did not see it that way, and the argument between them, which would eventually play out on a global stage, would become one of the most bitter priority disputes in the history of science. But that argument was still in the future. In August 1921, there was a more immediate problem. Banting's substance extracted from dogs worked. Now he needed to know if it worked in humans. And to find out, he needed it to be purified. Purified to a standard that would not kill a human patient with contamination before the substance itself had a chance to work. That task fell to a biochemist named James Collip. And what happened next, what James Collip did and then undid and then did again over the course of a single terrible week in December 1921, is one of the strangest chapters in the history of any scientific discovery. James Collip was brought into the project in December 1921 by MacLeod. He was a biochemist from the University of Alberta, visiting Toronto on sabbatical, and he was, of the four men now involved in the project, the one most technically equipped to solve the purification problem. Banting could extract the substance. Best could run the chemical tests. MacLeod could direct and interpret. Collip could purify. And purification was everything. The crude extract that Banting had been injecting into dogs was too contaminated for human use. It would trigger severe reactions. It might kill before it could treat. Collip spent weeks working through different purification approaches, adjusting temperatures, adjusting concentrations, testing batches on diabetic animals and measuring the results. And then, in the last days of December 1921, he found it. The exact combination of conditions that produced a purified extract stable enough, clean enough, consistent enough for human injection. He had the formula. And then within days, he lost it. Not the written formula, not a notebook, the knowledge. The intuitive understanding of exactly what temperature, exactly what concentration, exactly what sequence of steps produced the result he had achieved. Collip later described this period in correspondence that is preserved in the University of Toronto archives as a state of complete inability to reproduce what he had done. He knew what the end result should look like. He had the test data from the batch that had worked. He could not replicate the process that had produced it. For approximately 3 days, the accounts vary, the purification formula for insulin existed only as a memory that its creator could not access. The discovery that was going to save the lives of every diabetic patient in the world was temporarily unreachable, in the hands of a man who had forgotten how to find it. What happened in the laboratory during those 3 days is documented in accounts that differ on the details but agree on the atmosphere. Banting, by his own account, became physically threatening toward Collip. He described grabbing Collip by the collar, demanding to know what had happened to the formula, whether Collip, who was after all an outside collaborator with his own career interests, was planning to take the discovery somewhere else. The accusations were not made in writing. They were made in a small laboratory room late at night by a desperate man who had spent months working towards a single goal and was watching it slip away through a biochemist's lapsed memory. Collip denied withholding anything. He said simply that he could not remember. The formula came back, gradually. Over those 3 days, Collip retraced his steps, reran his tests, and eventually, whether through recovery of memory or systematic reconstruction, found the conditions again. The purification was re-established, the project moved forward. And on January 11th, 1922, a 14-year-old boy named Leonard Thompson was admitted to Toronto General Hospital, weighing 65 pounds. He was dying, and Frederick Banting had something to inject him with. The first injection of insulin into a human being did not work. Leonard Thompson had a severe allergic reaction within hours. His condition worsened. The injection that was supposed to save him had nearly killed him instead. Collip went back to the laboratory. 12 days later, on January 23, 1922, a second injection was prepared, a better purified batch, a refined formula. They injected Leonard Thompson again. Within hours, his blood sugar began to fall. Within 24 hours, it had normalized. Leonard Thompson, 14 years old, 65 pounds, days from death, woke up the following morning and asked for breakfast. He ate it. He digested it. His body processed it. For the first time in years, his body processed food normally. And standing outside the hospital room, not together, not speaking, were 4 men who had just changed the history of medicine, and who were about to spend the next 2 years trying to destroy each other. The news of Leonard Thompson's recovery spread through the medical community with a speed that, in 1922, was unusual. Word of mouth between physicians, letters, the medical grapevine that connected hospitals across North America. What had happened in Toronto was, depending on who was telling the story, either a miracle or the logical culmination of years of careful scientific work. The difference in how it was described depended almost entirely on who was doing the describing. And who was doing the describing depended almost entirely on who stood to benefit from each version of the story. MacLeod, as the senior scientist and laboratory director, presented the work at medical conferences as a collaborative achievement of his laboratory, with himself as the guiding scientific intelligence behind the project. Banting was furious. In his account, in his letters, in his later memoir, in his public statements, MacLeod had contributed nothing to the discovery itself. He had been in Scotland. He had lent a laboratory. He had returned, recognized the value of what Banting had done, and inserted himself in the credit structure in ways that Banting considered not just unfair, but deliberately deceptive. The argument was not private. It became, in the way that disputes between scientists sometimes do, a matter of public record. Letters were written. Accusations were made through intermediaries. The University of Toronto administration found itself managing a situation that had no clean resolution. Because the truth, the actual historical truth of what had happened in that borrowed laboratory between May and December 1921 was more complicated than either Banting or MacLeod allowed for. MacLeod had provided the resources. He had brought in Collip. He had directed the expansion of the project after his return from Scotland. He had, in the final months before the Thompson injection, contributed substantially to the scientific framework that made the work credible to the broader medical community. None of that was nothing, but none of it was what Banting had done in those first 4 months alone, in a borrowed laboratory, with a second-hand centrifuge, and 14 dogs. In October 1923, the Nobel Committee announced its decision. The Nobel Prize for Physiology or Medicine for 1923 was awarded to Frederick Banting and John MacLeod. Banting, upon hearing the announcement, was not grateful. He was publicly, explicitly, deliberately furious. He issued a statement the same day. He said that the award to MacLeod was an injustice, that the real collaborator, the man who had actually been in the laboratory, was Charles Best, and that he, Frederick Banting, was sharing his prize money equally with Best. MacLeod, upon hearing Banting's statement, issued his own. He said he was sharing his prize money with Collip.
2 Nobel Prize winners, publicly refusing to acknowledge each other, each one sharing their prize with the man the other had excluded. The Nobel Prize ceremony of 1923 featured 4 men with a claim to the discovery and 2 with the prize. And somewhere outside that accounting entirely, in a laboratory in Bucharest, Romania, a 5th man was reading the announcement. A man who had published his findings 6 months before Leonard Thompson received his first injection. A man whose name appeared nowhere in the Nobel citation. a man named Nicolae Paulescu. And what the Nobel Committee decided about Paulescu and why is a question that the historical record answers, but does not resolve. Nicolae Paulescu was a Romanian physiologist at the University of Bucharest. In April 1921, before Banting had extracted a single sample from a single dog, Paulescu injected a diabetic dog with a pancreatic extract he called pancreatine. The dog's blood sugar dropped. He published his findings in a Belgian medical journal in August 1921. The paper was peer-reviewed. It was accepted. It was in print. And in 1923, the Nobel Committee reviewed Paulescu's paper. They read Paulescu's paper. They acknowledged that his findings predated Toronto, and then they awarded the prize to Banting and MacLeod anyway. The official reason given was that Paulescu's extract was insufficiently purified for human use, that what he had demonstrated in dogs could not yet be applied to the clinical treatment of human patients. This distinction between animal proof of concept and human clinical application is the boundary the Nobel Committee drew. It is a defensible distinction. It is not a universally accepted one. And what Paulescu believed about why he was excluded, what he wrote in the years following 1923 in increasingly bitter correspondence with colleagues and in published articles, takes the story somewhere that the historical books almost never follow. Because Paulescu's explanation for his exclusion was not scientific, it was personal. And understanding it requires understanding something about Paulescu himself that the story of insulin almost never mentions, and that the Nobel Committee, in its official deliberations, did not address. Nicolae Paulescu was born in Bucharest in 1869. He was a physiologist, a scientist who studies the functions of living systems. And by 1916, he had been working on the problem of the pancreatic secretion for several years. The First World War interrupted his research. Romania was occupied. His laboratory was damaged. He resumed work in 1919. By April 1921, he had a functional pancreatic extract that lowered blood sugar in diabetic dogs. He called it pancreatine. He submitted his findings to a Belgian medical journal, Archives Internationales de Physiologie, in June 1921. The paper was published in August 1921. The Toronto team's first successful dog experiments were in July and August of the same year. Paulescu was first in print. The Nobel Committee reviewing the 1923 prize read his paper. They noted it. They acknowledged that his findings predated Toronto's publication, and they concluded that his extract, pancreatine, was insufficiently purified for clinical human application. That the critical step from animal proof to human treatment had been taken not by Paulescu, but by Banting, Best, Collip, and MacLeod. This distinction is scientifically defensible. The first human injection in January 1922 was Toronto's work. The clinical infrastructure that allowed insulin to be produced at scale was Toronto's work. The Nobel Prize decision, on its stated scientific grounds, has a logic to it. But Paulescu did not accept that logic. And what he wrote in the years that followed, in letters to colleagues, in published articles, in correspondence that is preserved in Romanian medical archives, suggests that he believed his exclusion was not primarily scientific. He believed it was anti-Semitic. Paulescu was a committed anti-Semite. He had published anti-Semitic writings. He had associations with far-right Romanian political movements, and he believed, stated explicitly in his correspondence, that the international scientific establishment, which he characterized in the anti-Semitic language he used throughout his career, had deliberately excluded him because of his political views. This claim is historically documented, and it is also historically complicated, because Paulescu's anti-Semitism is real and documented, and the Nobel Committee's reasoning is stated in their records, and both things can be true simultaneously. A man can have done genuine science and also be a person of genuine moral failure, and the history of discovery does not always resolve cleanly into heroes and victims. What is not complicated, what is not disputed, is what Banting did with the discovery after the Nobel Prize, and what the pharmaceutical industry did with it after Banting was gone. Because Banting did something with his patent that no medical discoverer before him had done. And what happened to that decision, what the market did to it over the following century, is the part of the story that is still happening right now to people who are alive today.
In January 1923, Frederick Banting, Charles Best, and James Collip assigned the patent for insulin to the University of Toronto for $1 each. 3 patents, $3 total. Banting's stated reason, recorded in interviews and correspondence, was that insulin had been discovered in a publicly funded university, that the patients who needed it did not choose to need it, that a drug that kept people alive should not be a source of profit for whoever happened to get there first. He wanted it available. He wanted it affordable. He wanted no pharmaceutical company to be able to corner the market on a substance that diabetics needed every day of their lives simply to remain alive. The University of Toronto licensed the patent to pharmaceutical manufacturers at minimal cost. For several decades, insulin remained relatively affordable. Banting died in February 1941 in a plane crash in Newfoundland en route to Britain during the Second World War. He was 49. He never saw what happened to his dollar. By 1996, a vial of insulin in the United States cost approximately $35. By 2001, it had risen to approximately $45. By 2013, it exceeded $150. By 2023, a standard vial of insulin in the United States, the drug whose patent was sold for $1 so that no one could profit from it, retailed for over $300 in many pharmacies. The 3 pharmaceutical companies that currently dominate insulin production in the United States reported combined revenues from insulin alone of over $14 billion in 2022. Banting sold the patent for $1. He is buried in London, Ontario. The grave is modest. 37 million Americans have diabetes. Of those, approximately 8 million require daily insulin injections to survive. Not to manage their condition, to survive. Without insulin, type 1 diabetics die. Not eventually, within days. In 1921, every one of them would have died. There was no treatment. There was the starvation diet, which extended life by weeks or months, and there was death. Frederick Banting changed that in a borrowed laboratory with 14 dogs, and until September. He sold what he discovered for $1 because he believed that a drug people needed to live should not be owned. That belief is documented. It is in his correspondence. It is in the public record.
It did not survive contact with the market. The insulin that costs $300 a vial today is chemically different from what Banting extracted in 1921. It has been improved, refined, engineered. The pharmaceutical companies that produce it argue correctly that the modern product required decades of additional research and development. They are not selling what Banting sold. They are selling what Banting's $1 made possible. That distinction between the discovery and what the discovery became is where the story of insulin lives today. In the refrigerators of 37 million Americans, in the insurance negotiations of people who cannot afford $300 a month, in the graves of the people who died in 1921 because Banting had not yet had his idea, and in a $1 patent that is now worth $14 billion a year. The archive does not close on Frederick Banting's grave. It asks what he would recognize. Subscribe to Hist Discover. The next file is already open.